Welcome back to another unit on evolution. This is a perfect opportunity to fuse what you've learned about adaptations in our previous evolution units along with our heredity unit. And so we're going to be focusing on evolution in terms of gene frequency. If you remember from our heredity unit, we looked at alleles, which referred to a dominant allele and sometimes a recessive allele, and when we have the two present together on homologous chromosomes within an organism, you have a heterozygous organism which expresses the trait that is determined by the dominant allele. Well, up to this point, you've learned about adaptations in terms of physical traits, and you've learned about genes in terms of alleles. So now we're just going to fuse them together and talk about adaptations in terms of how often you can find these alleles within a population. We call that gene frequency. And gene frequency, again, is just simply how often these alleles, capital A's and lowercase a's, can be found within a population. So to calculate how often an allele appears within a population, we're going to travel back in time and take a look at two guys, one by the name of Hardy and another mathematician by the name of Weinberg. The two guys collaborated together and derived an equation called the Hardy Weinberg principle to help us calculate how often these alleles, capital A's and lowercase a's, appear within the population. So let's jump right in. The Hardy-Weinberg equation is p squared plus 2pq plus q squared equals 1. Well, that's kind of complicated and meaningless, so let's go ahead and start with something that you can understand first. Let's imagine that we have a population, and within this population we have a variation of traits. This trait happens to be blue eyes, so we're going to say that this individual and this individual and this individual, 4 individuals have blue eyes, as opposed to the recessive trait, which is green eyes. Well, it's kind of difficult to tell how many alleles are capital A's and lowercase a's because the blue eyes could have the genotype capital, which is equivalent to homozygous dominant, or they could have the heterozygous form of the genotype, capital A, lowercase a. And the reason why they only express the capital A is because the capital one is dominant and the lowercase is recessive. Well, of course, that means that the green-eyed organisms have a genotype that is called a homozygous recessive, lowercase a. Well, a gene frequency, again, is simply saying, let's go ahead and count how many capital A's are present within the population. So there's 1, 2, 3, 4, 5, 6 capital A's. So we say that there are 6 capital A's, and we can count how frequently the lowercase a's appear within the population as well. That's 1, 2, 3, 4, 5, 6. There happens to be another 6 lowercase a's as well. If we add those together, you end up with a total of 12. And so we say that 50% are capital A's, which we represent by 0.5, and 50% are lowercase a's, which we represent by 0.5. We add those together, and we get a total of 1.00, which is really 100%. Well, in terms of genetics, the Hardy-Weinberg equation doesn't always work with A's. So instead of A's, we're going to use P to represent capital A and Q to represent lowercase a. Well, if we add these together, and again we had 0.5 P's and we had 0.5 Q's, then same thing, we add those together, P plus Q is 100%. 100% of the organisms within this population are accounted for now, and half of them, half of the alleles altogether are P's, and half of the alleles are Q's. So using the same 6 individuals, we can take them, we'll shrink them down here, and let's say that we take their genes and scatter them. So the alleles no longer appear in pairs, but they are all singular. And so we call this a gene pool. This is simply a pool of all the alleles that are present within the population. So from this gene pool, let's try to determine what the genes are going to be of the next generation. So what are my chances of pulling out a capital A? Well, there are 6 capital A's out of a total of 12, so that's 1 out of 2. And what are the chances of me bringing up another capital A to give me a homozygous dominant organism? Well, that is another 1 out of 2. And if we use our mathematical laws of fractions, then we know that the probability of pulling out a capital and a capital, we multiply those together, and that gives us 1/4. So the 1/4 of my next offspring, the next generation, will be the homozygous dominant for this trait of blue eyes. So let's do the same thing with the lowercase a's. So the green eyes, the recessive trait, what are my chances of pulling out a lowercase a? Well, that is 6 out of 12, which is equivalent to 1/2. And what are the chances of pulling out another lowercase a? And so we multiply that by that probability of 1 out of 2, and that gives us, again, 1 out of 4. That's 25% chance that I will get a homozygous recessive individual for this trait, which happens to be green eyes. So we've accounted for a homozygous dominant and homozygous recessive individuals, but we haven't done heterozygous yet. So let's do heterozygous. What are my chances of pulling out a capital A and then a lowercase a? Well, my capital A is 1/2, and my lowercase a would be also 1/2, and so that would give me a total of 1/4. So another 25% probability that I end up with a heterozygous individual. Well, there's nothing to say that I have to pull out a capital A first. I could pull out a lowercase a first. So my chances of pulling out a lowercase a, again, is 1/2, and my probability of pulling out a capital A is also 1/2. So we put those together, and we end up with another 25%. So in predicting the genes of the next generation, we have that 25% are going to be homozygous for blue eyes, and that's represented by 0.25, and 25%, 1/4, are going to be homozygous for green eyes, recessive, so that's 0.25 here. And in the middle, we have that both of these have to be added together. 1/4 plus 1/4 equals 50%, 25 plus 25%. So that's 0.50, that's 50%, are going to be heterozygous for this trait, capital A, lowercase a. So I know that was a lot. Stay with me here. We're almost done. So up here we return to, there was 50% probability that we pull out an A, and 50% probability that we pull out another A. So that really comes to A times A, which is A squared. Well, we come over to the right-hand side of the equation, and again, the probability of pulling out a lowercase a is 1 out of 2. And another lowercase a to give us a homozygous recessive gives us another lowercase a, so that's equivalent to lowercase a squared. So we've now accounted for what are the chances of giving a capital homozygous dominant, and we've also seen that there are two possibilities, two mechanisms in order to get a capital A, lowercase a heterozygous individual. And we've also taken a look at the recessive form of the trait to determine what's my probability of getting a lowercase homozygous recessive. So if we add all these up, the homozygous dominant with the heterozygous and homozygous recessive, then that gives us 100% of the individuals in the next generation. So that's how we derive the Hardy Weinberg equation. Instead of using capital A's, they decided to use a lowercase p, and so we represent that by p squared by substituting the lowercase a with a q squared. So a squared is now equivalent to the q squared. And the heterozygous form is equivalent to P, Q, and again, there were 2 of them. So we add that together, P squared plus 2PQ plus Q squared is equivalent to 1. So 100% of the next generation is the sum of all the homozygous dominant plus the heterozygous individuals and the homozygous recessive individuals of that generation. So now when we revisit the Hardy Weinberg equation, hopefully now it'll make a little bit more sense. We have P squared, which is equivalent to an organism with blue eyes, and the 2PQ, which also exhibits the dominant trait blue eyes, and we add that to our Q squared, which exhibits our green eyes, and in total of this next generation, we add the P squareds to the 2PQ heterozygous for the trait and the Q squareds, and that gives us 100% of the next generation of puppies with blue and green eyes. Now that we've represented visually the physical traits, let's revisit our mathematical equation. We said that, again, blue eyes is equivalent to P, and P was 0.5, 50% of the alleles. That's the allele frequency, and we said that Q is also equivalent to 0.5. We said that was the allele frequency of Q, and when we add those together, again, we end up with 0.5 plus 0.5 is equivalent to 1, so 100% of the next generation. We can apply that to the Hardy Weinberg equation and say that 0.5 times 0.5, because that's P squared, gives us a total of 25, or 25%. We apply it to the Q, again, was 0.5, 50%, times 0.5, 50%. That's the Q squared there. And that gives us the 25% probability that we end up with a puppy with green eyes. And in the middle, it was 2 times P, which is equivalent to 0.5, and Q, which is also equivalent to 0.5 from above there, and that gives us the total of the 0.50, 50% heterozygous individuals. And when we add those all together, then you can see 25% plus 50% plus 25% equals 100%. 100% of our next generation of puppies.